All right, I'm going to do a quick recording to go over a few of the AP questions on the pink sheets for this Unit 3 test. I did it in class with Period 3, but my screen wasn't sharing on the recording, so this is for Period 5 or anybody who is absent or anybody who wants to watch again. I did post this version, even though I'm not supposed to post. I'll probably delete it after the test, but the AP questions, and I don't know, it has some of my comments and stuff on it, so if it makes this easier. I'm only going to talk through a few of them that I feel like need explanation. First, skip number 2. We haven't done genes yet. Number 3, I did talk about with my Period 3 class, but I think I misspoke and said it was phosphorus that was lacking. And it says it does have phosphates, so that would be your phosphorus, but it's lacking nitrogen. Again, this whole thing is about sponge, and they often ask questions where, remember, the 6 elements found most in living things, and of all the categories, you sort of know which is which. So it says if you're lacking nitrogen, you know that proteins and nucleic acids have nitrogen, so they will go down if they're put in an environment lacking nitrogen. So that kind of question comes up a lot. Number 4, this I just wanted to point out, this is sometimes how AP asks questions in a weird way. They go backwards. They'll give you some sort of data or graph, and then they'll ask you, well, what do you think was the question? So just pick a logical question that kind of goes with what the graph is showing. Again, I don't think that's very hard, but just get used to that type of question. I'm not going to talk. I think you should be able to get all of these, but again, I posted this version with my comments. If you're not sure why the answer wasn't A, C, or D, I just made some comments there as to which part was incorrect for 7 or 8. I don't think 8 needs a lot of explanation, but again, often I try to write out, what they're telling you. So, we have this substrate, we have this product, we have ADH, which is the enzyme alcohol dehydrogenase. They talk about methyl alcohol as an inhibitor, so kind of draw out what the scenario tells you and then just pick the logical answers there. Number 9, you can see, I told you sometimes they'll have answers that are sort of true, but there's one that's better true. So number 9 is sort of an example of that. You can just see my comments there. Number 10, I wanted to point out, since we haven't done viruses, bacteriophage is a virus, but that infects bacteria, but it's a virus, and we haven't done viruses yet, but viruses have 2 parts. They're not a cell. They have a protein coat, which often can have different shapes. This one looks sort of like a spaceship with legs that land on the cell, the host cell. So this would be a bacteria cell if this is a bacteriophage. But viruses in general, whether they're bacteriophage viruses or not, they only have a protein coat and a nucleic acid. That could be DNA or RNA. They're not a cell with cytoplasm and organelles. And they land on a host cell that they infect, and we now know they inject. When we learn about viruses, you'll know they inject their DNA or their RNA into the host cell, which then has the code to make new viruses and take over your cell. All you really had from this one, even if it told you that they used a radioactive isotope of phosphorus, P32 is the one they use. We'll talk about this in a later unit, which shows up radioactively. You can detect where it is. And they used a radioactive isotope of sulfur, again, that you can detect where it is. Sulfur is going to label the proteins are going to be labeled with radioactive sulfur, and the DNA is going to be labeled with radioactive phosphorus. This is also just knowing that sulfur is part of proteins, but not DNA, and phosphorus is found in DNA, but not proteins. That's why they picked those 2 radioactive elements or isotopes. They then let it inject into the cell, and they tell you that they found radioactive phosphorus inside the cell, not radioactive sulfur. So basically, you're, again, just using the idea that phosphorus would be found in DNA, so it must be DNA that was injected inside. Number 11. All right, I like this one a lot, and it's a good thinking question. It gives you an example, sort of a story here of you have starch, which is your reactant or your substrate. Amylase is the enzyme that breaks that down into sugars, which we all know is glucose, right? Starch is made of glucose. Amylase is an enzyme that will break it down. We also have an indicator. So if we're trying to measure the rate of a reaction, you can measure the disappearance of the reactant in some way or the appearance of the product in some way. You have to have a way to measure or detect one or the other. In this one, they tell you we're going to measure the disappearance of starch. We have an indicator that's called IKI or iodine that in the presence of starch turns blue. So if we start out and if you have iodine and starch in a test tube, it'll turn blue. If there's no starch, the iodine by itself is like a goldish brown. So if we start out and we had starch in our test tube and we put iodine or IKI in there, it'll start out blue and it'll turn, and then you put the enzyme in there, the blue will turn brown as the reaction occurs. So that's how they're analyzing the reaction. If you read it, they're going to alter pH. I put this here just because I could ask any other type of question from a question like this. In this one, they're just asking you to justify what tube 5 is for. But I'd like you to think about this whole scenario because you see this one a lot. And sometimes they might ask you, well, what was the independent variable? What was the dependent variable? What do you think the null hypothesis would be? What would be some constants or controlled variables? So you should be able to tell me, the independent variable they're altering here is pH. You can see that in the first column. We're altering the pH in these different test tubes. The dependent variable is the rate of reaction. That's what they're measuring, and they're measuring that by the color change of blue to gold. The null hypothesis would be there's no significant difference in the rate of reaction at different pHs. Some of the constants, they probably are using the same tube, putting the same amount of substrate in, the same amount of enzyme in, the same this and the same, just all that stuff you keep the same. Anyway, what I wanted you to see up here, we have all these tubes. Our independent variable is pH. So the ones that I've highlighted here in green, those are the actual experimental groups where we're testing pH of 5, 6, 7, 8, and 9. Because in those tubes, you add substrate, you add enzyme, you add the indicator, which we need. And you don't boil the enzyme, which whenever you boil an enzyme, that's a way of having the enzyme in the tube, but it's denatured. So you'll notice that these, number 3, 4, and 5, these are negative controls. These are all at pH of 7. But remember, a negative control, you remove an aspect of the experimental setup, and it's called a negative control because you expect a negative result. It should not work. Nothing should happen. We just want to make sure that if I'm saying that as it turns brown, that is starch disappearing as it turns into glucose. Our method and that we're using, we're assuming all this is true that I just told you, but you have to have negative controls in place. So if you didn't put substrate in or starch in a tube, it should not turn from blue to brown if there's nothing there, okay? We have one tube with no substrate. We had another negative control where we did not add the enzyme, and another negative control where the enzyme is present, it's just not going to function. So those are a little bit different. One's the enzyme molecule's not in the tube, so nothing should happen. Another one is the molecule's in the tube, but it's just the wrong shape. So those are kind of 3 different negative controls, which is all they ask you about was number 5. But I wanted you to be familiar with those. You'll see some of those maybe on my test. Here's another one similar. In this case, if you read it, their independent variable is they want to see if these different bacteria strains are affecting this enzyme or this reaction. So again, you have a whole bunch of tubes where you added strain 1 and a whole bunch of tubes where you added strain 2. The ones in green are the true experimental groups where you put the bacteria in there You also have all the cellular contents, you have the substrate, and you have everything added in the tube. That's the true experimental groups in green. These other ones, again, are negative controls. Get used to looking at an experiment, picking out the negative control, or being able to give me a negative control. Here they just didn't add the cell contents. Here they didn't add the substrate. Here they didn't add the enzyme. We expect negative results in those because those are negative controls. Same thing here. The other one that I thought I should explain, like some of these are easy. Oh yeah, so this one, number 21 through 24, I explained it real quick. This is an enzyme lab, all right? So we're gonna do the enzyme lab after the test. We're gonna use a different method of measuring the rate of an enzyme reaction. This is another one that they sometimes cover, so you might see these in AP questions. So in this example, so we're doing the same enzyme, the same reaction. We're just going to assay it in a different way. They're taking hydrogen peroxide as your substrate, catalase is the enzyme. These are the products. In this one, they tell you the way you're going to measure the rate of this reaction is by measuring the appearance of product, in particular oxygen. And so how do we measure oxygen appearance? They take a paper disc. So like if you hole punched a piece of paper, of filter paper, think of that like a little paper hole punch, and you soaked that in catalase, the enzyme. So the enzyme is on the paper. And then you have a beaker filled with your substrate, right? Hydrogen peroxide. Then you take tweezers and you put the enzyme-soaked paper disc down to the bottom. And as soon as it's in there, the enzyme and the substrate start reacting. The catalase and the hydrogen peroxide, it's going to start doing this reaction. And the oxygen will form little bubbles on this little paper hole punch thing, and it'll float to the top. So the faster that this reaction goes, the faster oxygen's produced, the faster the bubbles appear, and the faster this paper disc will float to the top. And so that's just kind of the method they're using. In this case, they're altering the amount of hydrogen peroxide or substrate. But what you got to understand to answer the questions is that the faster the time that it floats to the top, the faster the time, which the smaller number, right? This one had a faster reaction rate because it only took 10 seconds to float to the top. So the smaller number here is a faster rate of reaction. And just understanding that, I think you can answer or look at the rest of these. And I think those were the only ones that I really felt needed explanation. Skip number 26. It talks about something in ecology we haven't done yet, so that one won't come up. And the rest I think are pretty easy, multiple choice. The grid in, this one is real easy. Again, sometimes they just show you, they give you a graph and they're just asking you to read a number off the graph. You don't even really have to understand what's going on here. So that's just reading a number off the graph. That's a short answer to practice. This is a long answer, a 10-point question that was on the AP exam that is similar to the one we're going to do in lab. And so I might have already posted a recording explaining this, but here's the reaction. Here's the enzyme. They're measuring the rate of this reaction. And in this case, they're, again, altering the pH. They put the substrate in there. They put the enzyme in each tube, equal amounts, right, in each tube. And they're just altering the pH in each one. And the way that they're measuring the rate of reaction, in this case, they're measuring this is the absorbance at a certain wavelength of 389, which we'll talk about in lab, but it's a way to measure how dark brown or how dark a color it turns. And it talks about here that one of the products is going to turn brown. So a higher absorbance, you'll get the darker the brown, the test tube, which means the faster the enzyme is working. And so you just answer the questions here. Skip D, we haven't done genetic engineering or DNA yet. You won't know how to answer D. But this is just an easy one. It just asks you to do a graph. and talk about a control. So there's the answer to that. So that is it. I think you have all the rest of these answers.